this video, we will talk about section 18.3 through 18.6, and this is on the evolution of the genome. So how has the genome of life itself, all living things on Earth, evolved over time? And so the first thing we want to talk about is how does the size of the genome from one organism to the other compare? And the thing is in general, prokaryotes have a smaller genome. Each cell, their genome, is only about 1 to 6 million base pairs long, whereas eukaryotes are much bigger. For example, humans have a 3,000 million or 3 billion base pair long genome compared to just a 6 million. However, inside of eukaryotes, so among eukaryotes, there's not much of a pattern. What do I mean by that? I mean that things like fruit flies have a longer genome than humans, or things like cacti have a longer genome than humans. So, genome length is not equal to complexity. Genome length is definitely not equal to complexity. In addition, humans, amongst all of the genomes, have the lowest gene density. What I mean by that is, in, let's say, a 10,000 base pair long segment of DNA, they will have less genes than maybe a mouse or maybe a cactus. So humans and other mammals have the lowest genome density. We have a lot of introns in our genes, a lot of non-coding sections of DNA between our genes. So we have lots of introns. Lots and lots of introns. So, what exactly does that mean? That means that 98.5% of our DNA does not code for protein. It doesn't code for RNAs or tRNAs or RNAs or things like that. They're introns, or they don't code for proteins or something like that. They don't go through transcription or translation. Some of it, called pseudogenes, are old genes that don't function anymore. Old non-functional genes, or repetitive DNA. So multiple copies of the same DNA sequence, repeating over and over and over again. And the last thing is there are things called transposable elements in our DNA that account for all of that. What transposable elements are is genes that move. They can move around. They can move around in the same chromosome. They can move around to a different chromosome. They can jump around. They're kind of nicknamed jumping genes. Alright? And if this is a chromosome and we've got a transposon here, or maybe a allele will be our transposon, it could jump over here, or it could even jump here. And the way it jumps a lot of the times is it either physically causes crossing over to happen, it touches, or it gets transcribed into DNA and it gets reverse transcripted via reverse transcriptase back into another chromosome. Actually, many scientists believe that the first retroviruses like HIV evolved or came about from transposable elements that encoded reverse transcriptase activity. So, about you as a human, 14% of your DNA is repetitive. What that means is it's just the same A, T, A, 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 G, A, B, the same sequence over and over and over and over again. So, for example, you could have something like GATA or GATG, and you could have that repeat 1,000 times. Or you could have something even shorter called a short nucleotide sequence or tandem repeat, like a G and a T, GT, that repeats 1,000 times. So, simple sequences of DNA contain many copies that tandemly repeat and short sequences that keep going over and over and over again. And the rest of your repeating DNA is something that is called multigene families. And these things are identical or similar genes that are repeated. So, some of these multigene families consist of exactly identical DNA sequences. Some are slightly different identical gene sequences. These multigene families, or families of genes that are identical or nearly identical, came about via DNA duplication. Remember when we talked about structural chromosomal mutations? A duplication on a chromosome has happened in the past to create these gene or these multi-gene families. A good example is in hemoglobin. There's alpha subunit of hemoglobin and beta subunit. They both occurred as part of the same gene family that do the same thing, carry oxygen. And they arose over the history of life because of mutation, because of chromosomal duplication, and because they imparted a favorable trait, they stuck around. What this brings about is a very important thing. The basis of evolution is mutation. Very important. Mutation causes evolution. The earliest forms of life had the least amount of genes, only the number of genes that they needed to survive. As mutations happen, duplications, inversions, things, the size of the genome increased. Accidents in meiosis can lead to extra and more chromosomes. Probably the first real life forms only had a few number of chromosomes. But over the years, as accidents in meiosis, polyploid, aneuploid events happen, extra chromosomes occurred, and these extra chromosomes caused the first speciation events, or species to diverge from the polyploidy. One great example is that humans have 23 chromosomes. Chimps have 24. What experimental evidence supports is that the chromosome 23 or 12 and chromosome 13 in chimps came together and actually fused together in human chromosome 15 over time. And we can see that because these ends of chromosome 12 and chromosome 13, those telomeres, when they merge together, we can see a telomere right here in chromosome 15. Telomeric DNA, remember, with those TAAAG, those repeats, those telomeres, we see that DNA in human chromosome 15. And that supports our hypothesis that that's what happens. So duplications, inversions, unequal crossing over during prophase 1 of meiosis can all result in chromosomes with deletions or duplications or things, and that increases genetic variability for evolution. In addition, transposable elements can provide sites for new crossing over, things like that to happen for evolution to occur. So things like meiotic crossing over, transposable elements, and duplications, inversions can all increase genetic diversity. Now, I understand one of the next questions will be, well, how does that relate to the creation of a new trait? For example. And what I'm going to do is use a specific example. So if we copy a gene, so let's say this is our chromosome. Here's gene A, and a mutation happens, and now we have 2 copies of A, and say A made some protein. Well, this original A is still making that protein. What that means is the second unneeded copy can mutate without endangering the function of this original copy A. So it can mutate, and a lot of the times it mutates into something detrimental, harmful, or it mutates into something that doesn't have any effect at all. But sometimes this can mutate and become beneficial. And that is the genetic basis of evolution. So there's quite a few protein coding genes that have multiple copies. And they arose by duplication and divergence. Okay? We can splice RNA different, create different exons and introns. And exons can be shuffled about and mixed and matched to create many different gene products. Transposable elements also have something to do with it. They can be inserted into different places in the genome and disrupt different coding sequences and block protein production and create different novel traits. Or they can also be inserted into a regulatory sequence that may increase or decrease a certain protein product. All right? So the last thing we're going to talk about is comparative genetics in evolution. So we can go on the basis of that closely related organisms have similar DNA. And the more similar your DNA, the more closely you are related. So we compare genomes of closely related species to help us understand the history of evolutionary events. So genomes that we compare of distant species can help us understand the past. And we can create what we'll talk about here in a while in evolution is phylogenetic trees that show how closely related A, B, and C are related based on genetic differences. So based on genetic differences. What we're going to notice when we compare genomes like that is that there are things called conserved genes. And those are genes that are the same in many forms of life. That means conserved genes evolved long ago. Many different organisms have these conserved genes. They have remained similar over long periods of time. They help us, these conserved genes help us clarify what the very earliest forms of life have had and where different genes began to diverge. So, the genomes of two closely related species are most likely to be similar, organized the same. Particularly, genetic differences between two species can be easily related to a phenotypic difference between them. So we can look at gene difference and try to correlate to a trait difference. And in this way, we can establish when different traits evolve in a family tree. For example, as a species of humans, we have about 200,000 years, and we have low intra-species, so in the same species, genetic variation. From human to human, our genes don't vary that much. Most of the variation within our human DNA is from things called SNPs, and those are single nucleotide polymorphisms, single nucleotides being different at different locations. And these variations can be very helpful in comparing lineage between different demographics of human beings. And kind of the last thing we're going to look at is evolutionary development. Evolutionary development. So embryonically, how do different species develop? Similarities in embryonic development require similarities in genes at the most basic level. So, we talked about earlier homobox or Hox genes, homobox genes. And these are genes that develop the body plan. So here's going to be a head, here's going to be a tail, here's going to be an eye, here's going to be, and we find very identical or near similar nucleotide sequences in these genes of both vertebrate and invertebrate. Homobox genes are very old. One of the very first genes, sequences, or families to develop. So, genes between us and flies have remained near the same since we diverged from fruit flies. And these homeobox genes that are in charge of regulating how the different parts of the body develop have stayed near the same. Same ones have been found in yeast or in plants. Many of those homobox genes encode transcription factors. So those that run transcription, that help us transcribe genes at specific locations. So what we can do is we can compare the DNA sequence or the amino acid sequence of an individual A, B, C, and D, and maybe A, let's say each of these letters is a different amino acid. Maybe it's C, F, G, D, A, C. Maybe it's C, F, G, D, B, A, C, C, F, D, B, C, G, F, and C, F, G, D, B, C, C. We can compare each amino acid or each nucleotide or amino acid. We can look for the differences, and as we compare them, we can see the differences between each of them, and we can see how closely two organisms are related based on their similarities and differences. So we can see that A and B are probably the most closely related. So if A is the parental species, B probably branched off of A, and D probably branched off of B, and C probably is split way out there. So we can compare relative to each other and their difference in genomes or sequences, how closely they are related to each other. How closely are they related to each other, and create a history of genetic events. All right. Now that was a lot, but we are going to go over this in some detail in class, and we'll do several activities on it so we can get started. This is the very basis of genetics. We can call this the evolutionary basis of genetics. So let me know if you have any questions. Thanks.